Good morning, everyone. It's a pleasure to welcome Antoni Ribas back to Stanford, friend and colleague and giant in the field of tumor immunotherapy. Tony got his early upbringing in his home country of Spain, where he got his MD PhD, the University of Barcelona. He did his internship and residency there, and then came to UCLA for a fellowship in hematology oncology and a postdoctoral fellowship. He was appointed to the faculty at UCLA in 2001, and he's now risen through the ranks. He's now the professor of medicine and surgery and molecular medical pharmacology at UCLA. He's director of the Tumor Immunology Program at the Jonsson Comprehensive Cancer Center and director of the Parker Institute for Cancer Immunotherapy at UCLA. His research is focused on laboratory and clinical studies, addressing the primary and acquired resistance to immunotherapy and targeted therapies, particularly in melanoma, and the development of gene-engineered adoptive T-cell transfer therapies for cancer. He's been instrumental in the clinical development of several agents approved by the FDA, including the first anti-PD-1 pembrolizumab, two combinations of BRAF and MEK inhibitors, and he helped develop the first CD19 CAR T-cell therapy for lymphoma, Yescarta. He's the immediate past president of the American Association of Cancer Research, AACR, and he's an elected fellow of the AACR Academy, the Royal Academy of Medicine, Catalonia, the American Society of Clinical Investigation, and the National Academy of Medicine. He has a very provocative title this morning: When the Cancer Becomes the Enabler of an Antitumor Immune Response. So, Tony, take it away, and everyone can ask their questions. Is put it in the Q&A section of the Zoom. So, Tony, welcome to Stanford.
Thank you very much, Ron, for this very kind introduction. It's always a pleasure to interact with you and your colleagues at Stanford. What I'm going to be presenting is data that we've generated recently, and most of it is published. But I've been trying to think, what is the real meaning of what we're seeing? Why does it work this way? And that's what I'm going to try to go through. It's going to be less data and more thinking. I'll start with my disclosures. I consulted with large and small pharma. I'm in the scientific advisory board of an increasing number of companies. With virtual meetings, it makes it more sustainable, but we'll see what happens when we go back live. And so please take this into account as I present this data. So I've been studying how patients respond or do not respond to immune checkpoint blockade with PD-1 antibodies, or with or without CTLA-4 blocking antibodies, here with nivolumab and ipilimumab. And by studying significant number of biopsies, then we can start looking at trends of what the data tells. We're looking for genes that lead to no response or increased response and things like this, and we've reported some of that. But there's no real smoking gun that says if you have this gene, you don't respond upfront or something like this, other than occasional losses of B2M or JAK kinases, which we'll go back to later. But if we take a series of over 100 biopsies at baseline and on therapy, and we follow them and we put them in a plot, and we put it in an artificial space called a principal component, where we let the data organize by itself. So there's principal component 1 and principal component 2. I'm trying to look for my laser pointer, but sorry, not this. I don't know, here. We can see that there's some outliers here. So each one of these dots is a biopsy. The open circles are baseline biopsies, closed circles are on therapy biopsies, and they're color-coded with progression disease in red, stable disease in green, and a partial complete response in blue. So we can see that there's these outliers. When you have a series of biopsies, some of them may be fibrosis or biopsied something else. But then we have all of these biopsies, the majority of them that have a directionality, and go from open circles to closed circles. So that means baseline to on therapy, and from red to green to blue, meaning no response, stable disease, or objective responses. And we try to see what is the feature that most aligns with this? It's actually driving a T-cell score or the genes that are associated with T-cells. And if we look at the same data in a different way, where we'll look at biopsies at baseline and on therapy with the same color coding, progression disease, stable, and complete or partial response, we can see that all of them go up, that the T-cell score goes up with immune checkpoint blockade therapy, even in patients who do not respond. It's just that the biopsies of patients who respond start with more T-cells and increase more. We know why this has to be. It makes sense, because if we're blocking PD-1, is because T-cells make interferon-gamma that leads to expression of 500 or so genes, one of them being PD-L1, and PD-L1 is used by the tumor to tell the cancer, by the cancer cells to tell the T-cells to stop attacking them. So if we give a blocking antibody to PD-1, then we're taking away this negative regulator, and we're hoping that all of the positive effects of the T-cells on the tumor mediated by interferon will go ahead. The data tells us that that actually happens. So here we have the biopsies that I showed in the principal component, but now each biopsy is a column. And here's we have a baseline and on therapy. Blue is negative gene expression, and red is positive gene expression. And we're looking for a series of genes that are interferon response genes. Interferon induces direct anti-proliferation and growth arrest in some cancer cells, not all of them. That's why it's called interferon, because it interferes with the proliferation of virally infected cells. And following with its role in fighting viral infections, interferon-gamma leads to the upregulation of antigen presentation machinery, HLA class 1 and 2, TAP transporters, proteasome subunits, and then it turns on interferon-gamma receptor itself and the signaling molecules, JAKs and STATs and IRFs, so it amplifies its own response. And then it makes a whole bunch of chemokines and effector molecules that attract more T cells. So what we see is that the giving of interferon turns on a program that changes the cancer cell. And if there's a disease progression, we see that on therapy, there's increase in red color compared to baseline. If we look at biopsies of patients with stable disease, we see that there's increase in red, meaning more of these genes on. And then we see patients with an objective response, they start with more of these interferon response genes turn on at baseline, and with therapy, they have even more. So if I turn this into a depiction, here's my depiction, which is a T cell sees antigen in the tumor, makes interferon-gamma that binds to the interferon-gamma receptor and turns on all of these genes. And now the cancer cell has become an enabler of the immune response as long as we block PD-1, which is what was turning off this and not allowing to amplify. Once this happens, then this one cancer cell that is being is turning on the interferon response genes starts being more visible to the immune system, makes it expresses more antigen, then it makes chemokines and cytokines that attract more T cells, changing the tumor microenvironment, which tells us that there's a key point here, that it's not only the T cells killing cancer cells, but the cancer cells helping amplify the anti-tumor immune response. And if we get it to this right level, that's when there's clinical responses. We see that there's evidence it happens in patients, in biopsies of patients who have no clinical response, probably because it's not ramped up enough so that enough cancer cells can do this. And it's not only in the interaction between the T cells and the cancer cells. It's not a one-on-one thing like the killing of T cells to cancer cells by perforin, granzyme, or fast trail and TNF is a one-on-one interaction. But this paper from Don Schumacher tells us that you can find interferon signaling up to 40 layers into a tumor. He did that by putting an interferon-gamma reporter gene into tumors and then inducing an immune response. So even really far away from where the T cells are, the cancer cells now have changed and start helping the immune response when everything works enough. which then made us think, so what if the biopsies of patients who do not respond is because they have an inherent lower ability to sense interferon and respond to it? And then once the biopsies of patients who respond to clinically, it's because they make more of these interferon response genes. A hypothesis that can be tested rather easily if you take a panel of human cancer cell lines. Here, we took 58 human melanoma cell lines that we had established in the lab over the years and we had characterized in different ways. And we exposed them to interferon-gamma and do this for each of these 58 cultures, and then take RNA before and after, and we look for which genes have changed, focusing on interferon response genes. And I asked my postdoc, Mykola Onyszczenko, to do 58 human melanoma cell lines because my hypothesis was that there was going to be different gradations of response, and some tumors may make a set of interferon response genes and others may make others. That's what I thought. Well, I was completely wrong. We could have done it with just 2 cell lines. The one that would represent the majority, which is the bulk of these 48 of the cell lines, where give interferon gamma and turn on the same genes. This just at 6 hours. If we wait 12 hours, some of the other genes may be turned on. And then, so these are the majority, telling us that the majority of at least melanoma cell lines have interferon gamma receptors and signaling an ability to make the same genes when exposed in an experimental way to the same amount of interferon gamma. And then there's the group of cell lines that did not respond. And we knew they would not, they were not going to respond because there were either naturally occurring melanoma cell lines that had JAK1 or JAK2 loss of function mutation, or model cell lines where we had CRISPR JAK1 or JAK2. And that's relevant because JAK1 and JAK2 is the bottleneck of the interferon signaling pathway, because if you lose one of them, either one of them, then there's no signaling. You do not need to lose both of them. Here I'm depicting both, but it's either of them will then lead to the cancer not responding and making all these genes that help the immune response. And why was this relevant? Because in data that we had generated before that had pointed us to the key role of the interferon gamma receptor signaling came from studying patients who had responded to anti-PD-1 therapy and after a period of time had progressed, what we call acquired resistance. When studying acquired resistance, you have a really controlled experiment. You have baseline, on therapy, and relapse tumor from the same patient. And we focus on very few patients, but we focus on the ones that we thought would be more informative, where the baseline and the progressive tumor, baseline and progressive tumor were the same lesion. So there was no clonal evolution in somewhere else. And these were patients who had responded for 1 or 2 years. I thought they were going to respond forever, and I was wrong. They progressed. It happens in around 1/3 of patients with metastatic melanoma that originally respond to anti-PD-1, also to anti-CTLA-4. With ipilimumab, we see exactly the same rate of relapse, just that there's numerically there's a lot less because there's less responses to anti-CTLA-4. But when we compare this tumor and this tumor, and we make a circus plot where we have chromosome 1, 2, 3, 4, 5, and go around, and we look for the mutations that are shared, which are the gray, are the majority of the mutations. 95% of the mutations in this tumor are the same as this one, telling us that the relapse tumor is very similar to the baseline. We looked for lost mutations, but they were mostly when the part of a chromosome was lost. That could have lost an antigen. We tried to convince us that that was the case. We couldn't demonstrate it. But then we looked at new and homozygous mutations, because this would be something that's really hard for the cancer to do, to have 2 alleles that were wild type at the beginning and then mutate one and make it homozygous. And if we look at this tumor, the homozygous would be in the virion allele frequency of 1, so this outer rim. And here's a red dot. I hope you see it. And then we go around and there's not other red dots. So there's only one new and homozygous mutation in this tumor that was not here. And that was a JAK1 loss of function mutation. Then we look at compare this one and this one, and we'll look around on the homozygous mutation. If it's new, a red dot, we go around, and there's only one here. We go around, and there's not another one. And this other one was a JAK2 loss of function mutation. So either one of the two kinases that are critical for responding to interferon-gamma receptor. And the way they happen is by a process called copy number neutral loss of heterozygosity, where there were two baseline wild-type alleles. One of them was lost, one of the wild-type alleles was lost. The other one was mutated with a detrimental mutation and amplified. So this is a loss of function of these kinases. Others since then have reported the same. Annette Pashin had a series of tumors from patients on immune checkpoint blockade and IL-2 therapy and reported all of these JAK1 and 2 and STAT mutations. And if we go back to the cartoon that we started with, here we're blocking PD-1 continuously. The T cells are making interferon-gamma. The cancer cells amplify the response. That leads to the clinical shrinkage of the tumor because now the cancer is scalping the T cells. It's not only a one-on-one game, it's that the tumor amplifies the response until the cancer, for whatever reason, figures out that, what if I stop doing this? What if I stop helping the immune system? I mutate JAK1 or JAK2, the only one gene that I need to mutate to become resistant, and then the tumor can start regrowing. So that's pretty powerful, because then we can think, if the tumor at these extreme cases, they're not common. There are probably around 10% of the cases that have acquired resistance. We found these JAK1 or JAK2 loss of function mutations. But it tells us a lot about how biology works, because it's a very controlled experiment and it's a clinical endpoint that was first response and then progression and a progressive disease. So if it all goes through the ability of the cancer cell to sense interferon-gamma and make interferon response genes, because that's what's lost when there's no JAK1 or JAK2. So we go from here, the T cells that make interferon-gamma, and they signal through interferon receptor, JAK1, JAK2, and STATs. Now, if we don't have JAK1 or JAK2, then the T cells still can see antigen, but the tumor cell no longer amplifies the response, and the cancer grows progressively. Since during the pandemic, we spent more time in our computers, I've mastered my bio-render skills, so now I can make these nice graphs. So here, I have a graph of what we thought we could do in this case. So if the cancer cannot make type 2 interferons because it has these loss of function mutations, but the tumor microenvironment still has cells that could make interferons. There's actually one type of cell, the plasmacytoid dendritic cell, which cell per cell makes a lot more interferon than any other cell in the body. And there's rare cells that are inside the tumor. So we thought, what if we inject a pattern recognition receptor that triggers type 1 interferon production by these plasmacytoid dendritic cells? Things that Ron Levy has published on intratumoral therapies that turn on a type 1 response, allowing the cancer that is not making the interference that we needed to make it, because these specialized cells, non-cancerogenous, can make it. As Ron, we worked with this one CpG oligonucleotide called SD101. It turns out that our body has evolved to recognize foreign sequences of DNA or RNA that look like bacteria or viruses, and CpG oligos look like bacterial DNA and some viral DNA. Double-stranded RNA looks like a virus. And our body has these toll-like receptors and other pattern recognition receptors that recognize these sequences. And once that happens, it senses the body that's under attack by a virus or a bacteria, and then makes type 1 interferons. So it induces all of these genes that are good for an immune response against the virus, but also against the cancer. So we tested this in a mouse model. We being my postdoc, Davis Torrejon, he established JAK1 and JAK2 loss of function MC38 colon carcinomas, which is a model of a high mutational load. I've been quiet too much and my lights turn off. It's a model of high mutational load colon cancer. And whenever we inject wild-type tumor, it grows progressively. If we give anti-PD-1, the red line, it gives an anti-tumor response. If we inject the CpG oligonucleotide, it's more response, and we give it in combination, it gives even more response. So now we're going to look at the mouse that has bilateral JAK1 loss of function tumors, and we're looking at the non-injected tumor. In the injected and non-injected tumors, the tumors grow progressively. If we give anti-PD-1 alone, there's no response. It requires JAK1 or JAK2. But if we give these toll-like receptor agonists, either alone or in combination, and in combination specifically for the distant tumors with anti-PD-1, we can get an anti-tumor response, telling us that even in the extreme situation of a genetic loss of function mutation in the tumor, we can get an anti-tumor response by inducing type I interferon production by these plasmacytoid dendritic cells. We studied all of these and published it in this paper. But what I'm going to go to is then going back to humans. So if this is true, then we can translate this approach and take patients with metastatic melanoma that are progressing on anti-PD-1 therapy. They have multiple metastases, they're not responding to the anti-PD-1, and we start injecting one of the peripheral lesions. In this time, we change to CMP01, which is CPGA TLR9 agonist. And then we continue this intralesional therapy plus anti-PD-1. And we've reported recently that when doing a series of patients that had progression on anti-PD-1, and we tried to change it by reverse the resistance by intratumoral injecting peripheral lesions, and we look at the response in all of the body, not only the injected lesions, we have an approximately 25% objective response rate, telling us that we can turn a quarter of the patients that have primary resistant metastatic melanoma into responding to anti-PD-1 by peripheral lesion injection with a CpG oligonucleotide. Here's one of these cases that was presented by Jason Luke at the ACR last year. I'm going to point this that here's a lesion, a leg of a person with a metastatic melanoma within transient metastasis that are progressing on anti-PD-1. Injecting just this one lesion, you can see that there's response in the injected lesion and the non-injected lesions. It would track CD8 cells, which would be the rare red dots here in the margin, increase with intratumoral injection, and not only in the injected lesion, but also increase in non-injected lesions. And PD-L1 also goes up because there's interferon production, which then tells us that we have to give this intralesional therapy plus an anti-PD-1 to offset the negative regulation. We've expanded on this concept because the TLR9 signaling is a potent one, but there's other pattern recognition receptors. And actually, whenever we look at the literature of recognizing double-stranded RNA, which has TLR3 and RNA helicases as the pattern recognition receptors, we saw that it did a feature that was very important to us, which is usually MHC class I expression and proteasome and antigen presenting machinery is under control of interferon. But if we take a tumor, well, there's the constitutive expression, but the upregulation is most frequently under control of interferon. But if we take a tumor that has JAK1 or JAK2 mutations, we cannot increase antigen presentation, even though we could get interferon signaling. So what if we can uncouple antigen presentation from interferon signaling and then get tumors to become more exposed to the immune system? So Anusha Kalbasi did this in a mouse model where he took B16 melanoma. B16 melanoma is notorious for being quite immune resistant. It does not express MHC class I at baseline. And only when we give interferon alpha, beta, or gamma, it expresses it. If it's a JAK1 knockout, then you give interferon alpha, beta, or gamma, and there's no increase on MHC class I expression. It doesn't go up this way. But if we give BO112, we do not get, we get MHC class I expression, but we do not get PD-L1, telling us that this double-stranded RNA can increase MHC class I without interferon signaling. And I'm sorry, I misquoted before. The MHC class 1 is in this axis. PD-L1, which is a surrogate for interferon signaling, is on the y-axis. So the key one is it's the difference between here that are double positive or here that only MHC class 1 increases. And that's because if you don't have JAKs, then you cannot signal through type 1 or type 2 interferon. And that not only happens in B16 melanoma, but if we take 1, 2, 3, 4, 5 human melanoma cell lines that do not respond to interferon, here we go from vehicle to interferon, they don't respond. To BO112, we have increase on MHC class 1, telling us that we can decouple interferon signaling from MHC class 1 presentation. And we think that that may be an added feature where going back to humans. And Anusha did a limited shRNA screen that was a while ago before we had CRISPR screens and shown that it was PKR and NF-kappa B and TLR3, the key components of signaling from these double-stranded nanoplex TLR3 agonists. And this has been taken in the clinic by a company in Spain called Highlight Therapeutics. And they've done the same that we did with the Checkmate Pharma study, take patients with metastatic melanoma that were progressing on anti-PD-1, and then they would get intratumoral BO112 plus continued anti-PD-1. And here, very consistent with the other data, there's a 27% objective response rate, telling us that we can rescue some patients with primary resistance to anti-PD-1 by the intratumoral injection of this double-stranded RNA with continued PD-1 therapy. They've also tested it in MSS colorectal tumors. Interestingly, there were no clinical responses, but on biopsies, they could show increase on CD8s and PD-L1, as we can see in this case and in this case compared to the baseline, telling us that you can, even in very low immunogenic tumors, you can change the tumor microenvironment by these intratumoral therapies. Probably in colorectal cancer, something else needs to be done besides bringing T cells and making interferon-gamma in the tumor. On the last part of the presentation, I'll go to the other mechanism of acquired resistance. This is another patient with who initially responded with pelvic lesions and then progressed in other places. Here's a circus plot again. We're comparing this tumor with this progressed tumor here. That's close to a year, 3/4 of a year later. And we're looking again for a new, so red dot homozygous mutation in the outer circle. We go around and around and around, and we don't find none until we get to chromosome 15, and we continue to chromosome 22, and there's no other one. So there's only one new homozygous mutation. This one new homozygous mutation was a loss of function mutation of beta-2 microglobulin. Was the same as with the JAK1 or JAK2. The wild type allele is lost, one of the wild type alleles, and the other one gains a loss of function mutation that makes the protein not stable, the RNA in the protein not stable. This has been replicated by Nir Hakomin and colleagues in patients with acquired resistant melanoma, and then by Katie Politi and Roy Herbst in patients with acquired resistant lung cancer, also showing that B2M loss is an occasional, rather rare, but validated mechanism of acquired resistance to anti-PD-1 therapy, and had been described by Nick Restifo as resistance to IL-2 20 years before. We can model this. We can take MC38, which responds partially. If we knock out beta-2 microglobulin, there's no response, suggesting to us that this is going to be a catastrophic event where you would think that the immune system would not be able to be reactivated, because beta-2 microglobulin is the required subunit for all the 6 alleles of MHC class 1 that we have. And if it's not there, then there's not going to be antigen presentation to CD8 positive T cells. And then how can this work? It cannot work because the T cells cannot recognize the cancer. The cancer now do not present antigens to the T cells. But we thought there would be a way around it, because CD8 T cells are not the only effector cells of the immune system. There's also CD4s and NK cells. So what if we try to supraphysiologically activate CD4s and NK cells, even though when CD8 cells cannot see their antigen recognition, their positive signal. Well, we thought we could do that by giving an IL-2 variant. And why an IL-2 variant? Because it's very potent in activating NK cells and CD4s. And actually, there's one that's called NKTR214. Now it's called Menpeg-L-dysleukin, which is a pegylated form of IL-2 that is pegylated in certain parts of the IL-2 protein and makes it more stable long-term. And actually, we have done studies in mouse models of adoptive cell transfer therapy, which I'm not showing you here, with imaging of the T cells, and we can see the mass expansion of CD8 T cells when we gave this pegylated IL-2 compared to native IL-2. So we thought that that could attract enough NK cells and CD4s to reverse resistance. So we go back to the same model, this MC38, that the wild-type tumor response in the red bar to anti-PD-1. We give the BEMPEG, the pegylated IL-2, it has a partial response like the PD-1. Give them together, we have even more response. If we take a B2M knockout variant of this MC38, now anti-PD-1, the red bar, no longer responds because the CD8 T cells, which are the main effectors, do not see antigen. But if we give IL-2 or the combination of IL-2 and anti-PD-1, we have a response. And if we deplete the different cell subtypes, if we deplete CD8s in purple, it does not take away the anti-tumor immune response, telling us that CD8s are dispensable in a B2M knockout MC38 to get an anti-PD-1 and IL-2 response. But if we deplete NK cells, CD4s, or especially if we deplete both CD4, CD8s, and NK cells, we take away the anti-tumor response, telling us that even though for I don't know how long I've been putting out these cartoons of a very simplistic interaction between a CD8 T cell and a tumor cell with MHC class 1 restriction and PD-1 allowing the CD8 T cell to attack the tumor, that it's not as simple, that CD4s and NK cells may also be mediating the response, at least in a model where we take the CD8s out. This agent has been developed in the clinic by a company called Nectar, which is in the Bay Area. Maybe some of you know the people there. And they have a trial that they reported on anti-PD-1 and NKTR214, suggesting that maybe there's more response in frontline therapy than if you would have with anti-PD-1 alone. I have not seen a study on patients who progress on anti-PD-1 giving the pegylated IL-2 and anti-PD-1, but there's frontline studies that are being done. So with this, I wanted to convey how I interpret how these therapies work. If a patient is going to respond to PD-1 blockade, it's because the tumor has enough T cells in there that are being blocked by just this one checkpoint. And once it's released, those T cells have enough strength to turn the tumor into an enabler of an immune response and make those 500 and so interferon response genes that change the tumor microenvironment, take away all of the negative regulators and bring in all of the positive cells. So you go from the M2 macrophages to the M1. Why? Because there's more interferon. You bring in more T cells. Tregs actually go up in the response, but they're not enough to turn it off because there's so many T cells, effective T cells, that are giving the response. And that's induced by a conserved interferon signaling pathway. It is really surprising to me that so many cancer cells, at least in melanoma, you give them interferon, they all do the same. They all turn on the same genes, unless they have JAK1 or JAK2 loss of function mutations. It's really interesting because there's very few cells in the body that make interferons. Interferon-gamma is only made by activated T cells and activated NK cells. But the majority of cells in the body have interferon-gamma receptors, and the majority of cancer cells have interferon-gamma receptors, telling us that it's not a coincidence, it's not an association, it's a causative event, because if a tumor loses the signaling, it can become acquired resistance. So that's why then we can think about it and say, well, how can we overcome resistance? We can do it by inducing interferon inflammatory response with the intralesional therapies that act on non-cancer cells that make type 1 interferon, or inducing NK and CD4s if what we lost was the ability of the CD8s to see recognize the tumor. The work that I presented to you has been funded by the National Cancer Institute, by the Parker Institute, Melanoma Research Alliance, ACR, and Stanford Cancer, CRI, and others. And here's my extended lab from before the pandemic. Now there's half of the people, and we don't meet and we wear masks all the time. So I'll be happy to take any questions.
So those of you who have questions, you can raise your hand with the icon at the bottom, or type it into the Q&A and I'll read it out. That's a really great summary of a lot of work over a number of years and really explains a lot of what we see in the clinic. So I'll start with the first question. The intralesional injection of CpG that induces interferon from the associated cells, the dendritic cells in the tumor. Why not just inject the interferon itself? Why induce an inducer of interferon when it's interferon that does the job?
Well, because it's been tried for years and it has little activity, both systemic and intratumoral, the same as injecting IL-2 systemic or intratumoral. There can be occasional responses, and the melanoma literature as well is well reported of local responses in injected lesions, but not in distant lesions. And I think it's because the key thing is to trigger physiological pattern recognition response, not only give the cytokine. So it has to be persistent enough that it can induce this amplification of the immune response and turning on and off T cells that have the right T cell receptor. I think cytokines have very poor pharmacokinetics. They disappear very rapidly. So it's probably not long enough and good enough. That's probably why the pegylated IL-2, I'm sorry, I should have put the data that demonstrates it, that's so much better than the wild-type IL-2 in activating T cells, because it's a longer kinetic exposure and triggering of the receptor.
So it's a depot or let's say a persistence issue rather than a mechanistic difference. I get
That's what I think. Others have looked at from this particular pegylated IL-2, the NKTR, that it's shielded from the alpha receptor of the IL-2 receptor, and that's, they say, well, that's it doesn't trigger Tregs, but we see it triggering Tregs and expanding. So I don't know how much of that is the mechanism.
Those of you on the line, feel free to ask questions. I'll ask a second one then. I'll ask a clinical question. You showed the role of JAK signaling, JAK1 and JAK2 in the cycle of events having to do with therapy with PD-1 blockade. We have people who are taking drugs that interfere with these enzymes for autoimmune diseases, various kinds. And even now we have JAK inhibitors that are approved for COVID infection. Is there any clinical observation about PD-1 blockade in people on these drugs?
Yes, so there was a company called Incyte that makes one of these JAK2 inhibitors that at the beginning of the development of anti-PD-1s, before we generated all this data, started a phase 1 trial of their JAK inhibitor plus pembrolizumab in patients with melanoma upfront. And they had to close it early because the response rate was so low that they were disappointed, and obviously makes sense. There's data from UPenn, from Andy Min, that maybe a delayed JAK inhibition while an ongoing, after inducing an immune response may be beneficial. The hypothesis being that continued interferon signaling may lead to turning off a whole bunch of pathways, and that you could use JAK inhibitors in a delayed fashion. In the Parker Institute meetings, I've seen some of the data of a clinical trial that they're open, suggesting that that may be an approach. I'm still concerned. I see the every time we looked at the biopsy of a patient responding early or late, there's interferon signaling. I don't want to turn that off with a JAK inhibitor.
Mm-hmm. Very interesting. I think we have a question from.
Yeah, so I can read it.
Can you read the question? Yeah, maybe you want to repeat the question as you answer it.
So could you speculate how the heterogeneity in the gut microbiome may fit into the framework that you outlined here? I'm not the right person to ask this question. I'm sorry, because we have not worked on the microbiome. I've seen the papers published. I've seen data from several groups saying that certain microbiome may modulate immune responses. We know that that's true in mouse models because we've all had bought mice from different colonies and had different immune responses to the same agents. But I don't know how, with the rest of the things that are required to have an anti-tumor response, I don't know how much the microbiome modulates it. Thinking about interference, maybe that it does, but I don't know if it's a causative event or something that's associated. So next question from Nathaniel Gray is, what are the prospects for immunotherapy in cancer prevention? These agents are being used in cancer prevention in patients with resected melanoma at high risk of relapse, both stage 3 and stage 2. If you give an anti-PD-1, the rates of relapse decreases. So that's cancer prevention in preventing the development of metastases after surgery. So that means that there were some dermal metastases that can regress by giving these therapies, which is not surprising. We always have to balance with the toxicities, but when it's anti-PD-1 alone, it's very in favor of the therapies, despite occasional patients having inducing autoimmune processes with the therapy. To prevent cancer in people who do not have cancer, I think that's going to be a harder sell because around 25% of the patients who receive anti-PD-1 alone develop hypothyroidism, which can be treated by giving thyroid hormones, but it's something that has to be done for life. Around 1 in 20 develop adrenal insufficiency. You have to give hydrocortisone for life. Others can develop pneumonitis, hepatitis that can be life-threatening occasionally. That's less than 1 in 20. But still, if we're thinking about prevention, I think it's going to be a class of drugs that probably are not going to be used unless there's a very high risk of a certain cancer in that population.
So I'll come to a third question then, and as we wait for others. As you know, the champion tumor responding to PD-1 blockade is not melanoma, it's Hodgkin's disease, where we have in range of 80% response rate to the single checkpoint inhibitor PD-1 blockade. And Hodgkin's disease is in the category of things that you presented because beta-2 microglobulin mutations are common in Hodgkin's disease. And the tumor doesn't have class 1 because it doesn't have beta-2 microglobulin, like the case you discussed in your model. And so why does Hodgkin's disease respond so well to PD-1 blockade?
And Ron, yeah, this is something we've talked before, and I'm glad you're bringing it up, because besides Hodgkin's disease, there's clear evidence, and we've also found it, that occasional tumors that have B2M loss at baseline in patients with melanoma or lung cancer, or even more frequently in patients with MSI-high colorectal cancer, so the high mutational load colorectal cancer, where there's accumulation of B2M loss of function mutations at baseline before doing immunotherapy, that occasionally some of those patients have objective responses. And I've been in meetings where I've heard, oh, Antoni Ribas is completely wrong because look, this patient responded despite the B2M. I think it's very different when you have it up front, where the immune system has evolved one way, or where you have it as the acquired resistance, where the only change was the loss of B2M. I think one thing does not take the other one away. A tumor that had B2M responds to PD-1 loses it. That is a causative event for resistance. But a tumor that has no B2M at baseline can still respond, probably because the PD-1 blockade may induce NK and CD4 responses, all poorly characterized. Alberto Bardelli has a paper in a mouse model where he shows CD4 T-cell responses. We've seen CD4 and NK cells. We're trying to study these rare cases of B2M loss at baseline that respond. And if I had thought more and I realizing that I had extra time, I should have put some of that in because it's very interesting biology. In the mouse tumors, what we see is that the B2M knockout tumors have less DC1s in them, which is surprising because it's the tumor that doesn't present antigen, not the dendritic cells. But the B2M loss leads to less DC1. When we give the IL-2, then we reconstitute that loss.
Yeah, I think those experiments speak directly to the Hodgkin's disease dramatic responses. It's probably CD4s and NK cells that do the job rather than the cytotoxic CD8 T cell, which is instructive because it shows that the PD-1 blockade story has many facets, not just the single diagram that everyone usually shows. So I think your work really illuminates so much of why this works so dramatically in so many cases. And when it doesn't work, why not? So I really salute you and thank you very much for amazing talk and amazing work. And we look forward to following your work in the future. Thank you very much.
Thank you very much.
Today.
Bye.